is the LB amp with no pGLO. So this is E. coli cells that did not get the pGLO plasmid. So they will not have the ampicillin resistance gene. So they will not be able to inactivate the ampicillin in the agar. So they die. So they did not grow. Okay, so that's that one. Here is regular E. coli, and you can see the boundary there. There's a ton of growth on this plate. So basically, just super, a ton of E. coli streaked everywhere, okay? This plate has normal E. coli. It was not given the plasmid, and it's on a normal LB agar plate. So it should grow abundantly, and we call this type of growth a lawn or confluent. It's confluent growth, just a lawn of bacterial growth. You can't even see colonies. It's so much. Okay, so there's that one. Then you have this plate, which is the LB amp arabinose. So this is E. coli that got the pGLO plasmid. So it should have resistant to ampicillin. So it should be able to grow, and you can see some colonies in there. So yes, there is growth, okay? Is there confluent growth? No, there's not a lawn of bacterial growth because you didn't get the plasmid into every single E. coli cell. But you do have success, you got it in. And then we give arabinose to serve as the inducer to induce that GFP gene to be expressed. So if we take our plate with all those colonies and we hit it with the UV light, oh, look what we see, popping green, okay? So this is our experimental plate. It did work. We are happy. We have transformed E. coli, yay. And then this plate is your LB ampicillin plate, okay? This is the E. coli that was treated with the plasmid, so they got it into their cells because look, you have some colonies. So it did grow. We have colonies. Why did it grow? It got the plasmid put into it. So on that plasmid, we have that beta-lactamase gene that will cut and inactivate the ampicillin. Takes out the ampicillin so it's able to grow. So this shows us our transformation was a success. But do they pop green when we hit them with UV light? No, they do not. And why do they not pop green? Because the plate does not have the inducer here, arabinose. Okay, so this is still an experimental plate, but it's different in that there's no arabinose. So these colonies will pop green because the arabinose inducer is there to switch on the GFP gene. These do not pop green, but they did grow because they're going to resist the ampicillin. Those are your two experimental plates. They worked. Good job, table 3. This plate is basically a negative control. The E. coli that did not get the plasmid was killed. So nothing grew. So that's exactly how it's supposed to be. Nothing should grow. The E. coli that didn't get the plasmid, just normal E. coli that we started off to make sure it is viable and can grow, is put on this plate. And did it grow? Yeah, it grew as a lawn. So we know that we had viable E. coli cells to begin with and that our experiment should go. Okay, so this is called the positive control. We know the bacteria can grow. We know it was killed with the ampicillin because E. coli will always be killed with ampicillin unless it gets what? Unless it gets the resistance gene, the beta-lactamase resistance gene found in pGLO. So those E. coli cells, those colonies are from one cell that got the plasmid. And then you had a lot more that also are gonna pop green once we give them arabinose. So good job, table 3. And that is the explanation of your transformation plates.